Assalamu alaikum students. Today our topic of discussion is a complement system. So complement system for antibody action. There are 20 proteins that are enzyme precursors. Principal actors include 11 proteins C1 through C9, B and D. All are in blood and tissue spaces activated by classical pathway. So the classic pathway initiated by an antigen-antibody reaction. So first of all, there is antigen-antibody reaction which activates the classical pathway. Antibody becomes uncovered or activated, and this binds with the C1 molecule of the complement system, setting a cascade of reactions beginning with activation of the proenzyme C1 itself. There are multiple end products. You can see this is the classic pathway. First of all, there is antigen antibody reaction, then there is activation of C1, and this C1 activated in the presence of C1 activated, the C4 combines with the C2 and results in the production of C42 and C4A. This C4A activates mast cells and basophils, while the C42 will lead to the activation of or the conversion of C3 into C3b and C3a. The C3b is important for the opsonization of bacteria, while C3a is important in the activation of mast cells and basophils. C42 and C3b both will convert C5 into C5b and C5a, while for the activation of C3 into C3b and C3a, there is also the role of B and D complement proteins. So the C5a is very important regarding the chemotaxis of white blood cells, while C5b will lead to the reaction in which C6 combines with the C7 and leads to the production of C5b6 7, and this complex will lead to the reaction in which C8 and C9 combines to produce C5b6 7 8 9. This complex will lead to the lysis of cells. So here you can see the different functions of complement system or the antibodies in killing in killing the foreign agents, and there is the role of antigen antibody complex in immunity. Lysis, a combination of multiple complement factors and designated C5b678 9 complex. This has a direct effect of rupturing the cell membrane of bacteria or other invading organisms. Agglutination, the complement products change surfaces of the invading organisms, causing them to adhere to one another, thus promoting agglutination. The other effect of a complement system is the neutralization of viruses by the complement enzyme and other complement products. There is the neutralization of viruses. So chemotaxis, fragment C5a initiates chemotaxis of neutrophils and macrophages, thus causing large number of these phagocytes to migrate into the tissue area adjacent to the antigenic agent. Activation of mast cells and basophils. Fragments C3a, C4a, and C5a activate mast cells and basophils, causing them to release histamine, heparin, and several other substances into the local fluids. Inflammatory effects. These substances in turn cause increased local blood flow, increased leakage of fluid and plasma proteins into the tissue and other local tissues, reactions that help to inactivate or immobilize the antigenic agent. The same factors play a major role in inflammation. The other effect of the complement system is the opsonization and phagocytosis. One of the products, C3p, activates phagocytosis by neutrophils and macrophages. So this is all about the complement system. You can see this is the complement system or the classic pathway, which include different complement proteins, and these are the functions of the complement system or the classic pathway, that is the lysis, agglutination, neutralization of viruses, chemotaxis, activation of mast cells and basophils, and opsonization and phagocytosis. So these are the different functions by which there is the destruction of foreign agent, or there is the paralysis of foreign agent. So you have to remember that which complement protein is important in which function. So this C3b is very important, and this C5a regarding chemotaxis is very important, and this complex is has an important role in lysis. So this is all about the complement system. If you have any query, you can contact me. Thank you.